Okay, so let's continue our series of lectures on quantum biology. One way of connecting physics to biology is through electromagnetic fields, and this has a long history, going back about a century to a Russian scientist, Alexander Gurevich, who introduced the concept of morphogenic fields in order to explain some synchronization of living systems on an organism-wide scale, and also linking it to the formation of structures and form by living systems, which otherwise is very hard to explain. And he proposed that there exist morphogenic fields, in principle electromagnetic fields, which extend beyond these structures, biological structures, into space and guide the evolution of these organisms. But there is more recent examples. There are more recent examples of the application of such large-scale concepts for biological systems outside of, let's say, structural biology and biochemistry. Two examples are shown in this slide, where I've included the work of Herbert Fröhlich, a very well-known physicist, who in the 70s and 80s proposed the existence of non-equilibrium states, which arise from long-range coherence, and so that's the concept. Biological coherence was the topic of his work, and that means that not individual cells, but cell assemblies interact and create a uniform physical state, which in his case, he proposed that this would be mediated by dipole-dipole interactions and high-frequency oscillations of biological dipoles, principally in the membranes of cells, and these become synchronized and give the entire organism a sense of unity. A more sort of mathematically grounded work was developed by Nobel Prize-winning chemist Ilya Prigogine in Brussels, and he made great progress in explaining how complex forms emerge from coupled nonlinear chemical reactions. And so there was the introduction of reaction diffusion kinetics for chemical reactions that resulted in heterogeneous structures, and a lot of applications followed, especially in the field of embryology, as well as mathematical biology, which was basically enabled by these kinds of nonlinear differential equations that were able to explain things like the pattern formation on the coats of animals, zebras and jaguars and so on, by modeling mathematically, very precisely, the reaction kinetics taking place in these systems. A few words, maybe briefly, about Fröhlich's long-range coherence, which is still discussed, and I think it has some relevance. So three major assumptions in this theory, and I'm mentioning this because of the electromagnetic concepts and actually the quantum mechanical foundations. So although at the end of the day these models can be understood by classical terms, but the foundations are quantum mechanical in terms of dipolar oscillations and at the level of atomic molecular systems. So the main assumptions that Fröhlich put forward in his theory was, number one, that there is a metabolic energy supply, or any sort of pumping akin to laser action, and that is very specific to biological systems, which I mentioned earlier. Otherwise, the system would not be able to achieve maintenance of a high energy metastable state, which is organized. Otherwise, it becomes disorganized and forces of entropy take over. The second aspect is the presence of thermal noise. As I also indicated earlier, thermal energy is provided for free, at kT amount per degree of freedom, temperature being physiological temperature, so by physical standards quite high. And therefore, this noise actually has to be accounted for, otherwise we'd be talking about completely hypothetical systems kept artificially at very low temperatures. And number three, and crucially, these dipoles interact through nonlinear coupling. So nonlinearity enables the system to find higher energy metastable state, as I said. And the prediction was based on the physical dimensions of membranes and the dipolar strengths that this high energy state is characterized by a frequency of about 10 to the 11, 10 to the 12 hertz, so low terahertz range. And that's interesting because these days I think we are developing a lot of terahertz technology, including airport scanners for security systems and also for imaging in medicine. This range of frequencies is being exploited, and we know that it's something to be aware of, and perhaps also somewhat cautious about the use of these kinds of frequency, which may have both positive and negative or destructive effects on living systems. If we are to believe Fröhlich, there is unfortunately not a lot of empirical evidence, although the theory was developed at a time that we didn't have this kind of technology that we do today, about 30, 40 years ago. The next topic that now brings us closer to living systems and actually to something which is demonstrable experimentally is the work of Günther Albert Bühler from Northwestern University in Chicago, and he spent about 20-25 years of his life as a scientist exploring cell-cell communication and positing or hypothesizing that this takes place through electromagnetic interactions between cells. And to this end, he coined the term intelligent cell or cell intelligence, which means that cells, single cells, individual cells, including neurons, which we'll be discussing later in this course, communicate with other cells by electromagnetic means. And of course, the question is now what frequency that would be. I mentioned earlier that infrared electromagnetic radiation is abundant due to finite temperature and irradiation of thermal noise. But in his case, he studied cells interacting at much higher frequencies and higher energies, which is therefore called non-thermal. And his website, called Cell Intelligence, explores in detail his findings and consequences. And you can say that through very skillful manipulation of the experimental apparatus and the setup, Günther Albert Bühler was able to demonstrate that there's definitely effects of cells perceiving other cells in their neighborhood and reacting to the presence, and he was able to determine that there is no either chemical or contact interaction between these cells. So more or less what's left is electromagnetic. If there's no chemistry, there's no signaling through chemical molecules or through direct contact between cells, mechanical, then the only thing that one can think of is electromagnetic. And then he used a very clever setup where beads were dispersed in the solution with living cells present, and these beads were able to scatter laser light of different frequencies that he used, and that indicated the effects on those cells. So when they perceived the presence of light in a particular wavelength of significance, they would be attracted to it. And the graph on the right shows basically the response intensity depending on the wavelength. And the wavelength that the cells recognized were ranging from near infrared, something like 1500 nanometers, to visible, down to about 600 nanometers. So, so definitely this is non-thermal. These are non-thermal frequencies. Some of it is visible, some of it is near infrared. And the question then remains, first of all, if that's the case, what is the emitter of radiation in the case of a living cells, and what is the receiver, and finally, how is it transduced into mechanical response? In other words, one cell has to emit electromagnetic radiation at a particular frequency, which would be then received by another one. In an analogy with an antenna, let's say, you have an emitter, an antenna in the receiver, and then this has to be transduced into, let's say, sound in the case of radio reception, transmission and reception. So that's the analogy, and he postulated and explained in great detail that the receiver is the centriole, which is one of the organelles in all the eukaryotic cells, composed of 9 triplets of microtubules, a very intricate arrangement, a very complex architecture, which involves, as I said, 9 triplets of microtubules in a central doublet. And he speculated further that the centriole can operate as a primitive eye, recognizing the source of electromagnetic radiation to within about 4 or 5 degrees in both the in-plane direction and out-of-plane direction, giving the cell fairly accurate information about the direction from which light is coming. And then we'll come back to it in a few minutes, because it makes more and more sense in view of recent experiments that actually these microtubules that form centrioles may in fact be able to not only absorb this kind of frequencies, but also maintain long-lived quantum states, electronic states that could be then transduced into mechanical action. So, but the question is, where is the light coming from? This is only what is receiving it. Albert Giller speculated it could be due to the mitochondrial activity. Together with my late colleague John Dixon, we wrote a paper showing that this is entirely feasible, that proton pumps in mitochondrial membranes, due to proton transitions through it, are able to emit radiation exactly in this frequency range that Albert Giller postulated. So that's a very interesting story. We'll come back to it. Cell-cell communication, interaction between cells, and beyond contact, beyond chemical or any other signaling molecules. There is corroborative evidence coming up in the case of chloroplasts, and we'll come to it in photosynthesis, where structures which are at the molecular level somewhat similar to those that we find in, for example, microtubules, are involved in absorption and funneling of electromagnetic energy into into specific sites that can collect it and then transduce into electronic transformations. So this is an example of a recent paper where green and blue light is involved in the chloroplast migration. And so there is definitely an example of electromagnetic transduction into mechanical movement in these systems. Okay, a somewhat related topic, and actually going on a fairly intriguing tangent, is the work of a group of scientists, radiation biophysicists from McMaster University in Hamilton, Ontario, led by Professor Carmen Mothersill. They study effects of electromagnetic radiation in living systems at all scales. Of course, specifically, we are interested in therapeutic effects of radiotherapy in cancer, for example, but they've also been experimenting with individual cells and different organisms, including schools of fish. And one of the effects in cancer therapy that has been quite well documented and is well known is the so-called bystander effect, whereby cells exposed to either radiation or chemotherapy and therefore receiving damage from the environment, from the therapeutic apparatus, for example, signal in some way to other cells which are not directly hit by these modalities, and those cells which are not directly hit actually respond the same way. So it's kind of that's a bystander effect. It's almost like the message is going out, we are being hit, run for cover, or express some protective mechanisms, DNA repair enzymes, or P-glycoproteins on the membranes of these cells. The damage is due to chemotherapy, and protect yourself. So these experiments are incredibly interesting because they show us that cells do communicate, perhaps directly, but also indirectly, because some of the range of these bystander effects is quite amazing, and also extends to full organisms, in which case we don't have a good explanation other than assume electromagnetic radiation used for communication. So this is an example of significant events triggering a response at the level of a community of cells, a community of organisms of the same species, let's say, but not only. And these signals then alert other members of that society of cells or organisms to prepare and repair, prepare for damage and repair damage. So that's the effect which actually extends beyond, sometimes beyond the organ which is targeted by radiation, or beyond the species. So some recent papers were actually quite intriguing, and I'll just describe, and you can look up the work of Professor Mothersill in Google Scholar or some other means. One of the more recent bizarre experiments indicated that in an aquarium with two groups of different species of fish, separated by a net, only half of the aquarium was exposed to radiation, and the other half was not. And the response in terms of, for example, heat shock protein activation was identical in both. Those fishes which were not exposed, they reacted the same way. A more bizarre even experiment was conducted where reaction to the unexposed animals to radiation delivered to the exposed animals occurred before the event happened. And that's in published literature, peer-reviewed papers. In other words, it's almost, it's some form of premonition or time travel, if you will, in terms of recognizing danger. Perhaps it's simply pattern recognition or some kind of memory. I don't know the details, but it definitely merits great attention. The authors claim that this involves quantum entanglement, which would be quite revolutionary if it was demonstrated to be true. And of course, a related question is how molecular level signals get amplified in the bystander effect to the entirety of the organism, which is exposed to these dangerous effects. And these are two of the more recent and more intriguing experiments from that group on bystander effects and the molecular level responses. Okay, I'm going to switch gears now and talk about something also which falls into the class of quantum biology effects, and perhaps the most elegantly and completely developed theory of quantum effects in biology by Lloyd Demetrius, who is a mathematical biologist, and he coined the term quantum metabolism, as opposed to classical metabolism, and I'll explain the difference in a few minutes. But again, this is grounded in a physical theory called quantum theory of solids, with a very illustrious history. Quantum theory of solids considers crystal lattices, and the original experiments on the behavior of solids exposed to temperature changes was to measure their heat capacity. That means how much thermal energy needs to be delivered to increase the temperature of the solid by a given amount of temperature difference. And it turns out, and this is a very well documented part of physics going back more than a century, that at low temperatures, close to 0 Kelvin, specific heat of solids, or heat capacity of solids, follows dependence on absolute temperature to the power 3, cubic dependence on temperature, whereas at high temperatures it goes to a constant value of the gas constant R. So that was the knowledge about a century ago already, and the question was, how do we explain this thermal dependence of specific heat of solids? Albert Einstein had a hand at this, and he actually failed, probably one of the very few cases where Einstein made a wrong prediction or incorrect model. The Einstein model assumed that solids are composed of atoms or molecules which vibrate independently, and he used basically Planck's model for black body radiation and adapted to a solid crystal, let's say. It turned out that his theory led to the correct behavior at high temperatures, namely constant value of the specific heat. However, at low temperatures had a very incorrect temperature dependence of the specific heat, namely exponential dependence on temperature, and that was not what was definitely established experimentally. However, Debye, a contemporary of Einstein who got the Nobel Prize for this correct model, assumed something else. He assumed that solids are composed, yes, of atoms and molecules, but these elementary entities forming a lattice are not vibrating independently of each other. They are actually forming waves of vibrations, and these waves have the different wavelengths, and the shortest wavelength would be 2 atomic distances, lattice, unit lattice values, and the largest half wavelength would be the size of the solid. And anyway, this gives the limits on the frequencies through this propagation velocity. And when that's included in the theory, and statistical thermodynamics is involved in the calculations through the partition function, the result is the correct behavior, and at both low and high temperatures, that explains the way solids transform thermal energy into movement. Vibrations in the form of waves propagating through the entire solid. That's very important because this is an idea of a coherent behavior of a large microscopic object. And in this case, actually, it's a quantum, so all these waves are quantized, just the same way as Planck quantized electromagnetic waves, except these are mechanical waves, not electromagnetic necessarily. Could also be if there's charges involved, but in the most direct way, they are just mechanical waves which are quantized because of the periodicity of the lattice. And furthermore, which is kind of a footnote to it, this t to the third power dependence is valid in three-dimensional solids. Let's say, think about a cube, but if the solid is considered to be let's say, a linear string of atoms or a plane composed of atoms, then the power changes, it actually goes down. And that's important because Lloyd Demetrius translated this model into biological context of metabolism. So how did he do it? Well, first of all, he drew on 2.5 centuries of investigations into metabolism of all species, but metabolic rates of bacteria, plants, animals, humans, and it's very interesting because there are a lot of regularities if you study this very carefully, and the story goes back to Laplace and Lavoisier in 1780, who, if you recall, this is the time that combustion engines were conceived and created and changed our society through the Industrial Revolution. So thermodynamics was born at that time, and these scientists, the physicists and the chemists, viewed the living system as a kind of thermodynamic engine. So combustion, so oxygen is needed for respiration, and respiration leads to, as you will see in a second, to metabolic energy transduction into biological quanta of ATP. So they got the right initial concept, that it is some kind of combustion. Then, more than a century later, Rubner started classifying the rates of combustion or energy transduction, i.e. metabolic rates, in domesticated animals, and relating them to body size and lifespan. So they found, Rubner found for the first time, scaling relations. In other words, various properties of living systems, including lifespan, the rate of metabolic energy production, and other properties, heartbeats, and so on, with scale of the system. And there are regular properties, which is quite amazing because in biology we have so much diversity, very little uniformity. And here actually is an attempt at finding mathematical relations for biological organisms across species and kingdoms even. Eventually, in 1940, Kleiber systematized all of this and found a mathematical relation between metabolic rates and body size. And here is kind of a brief summary of what this means mathematically. Y is some property, could be rate of respiration or heartbeat, or the frequency of heartbeats, or heart rate, I guess is the term, and or energy consumption, and W is the body size or weight, and alpha and beta are parameters to which you fit lots of data. So this can be measured by the respiratory cycle, cardiac cycle. As we know, all of biology is about cyclical or repetitive behaviors. Starting with circadian rhythms, we respond to the sunlight and lack thereof. And so there's shorter, faster cycles, and the fastest cycle is metabolic cycle, about a millisecond, as you will see in a few minutes, which is the time it takes to produce one ATP molecule from ADP. And you can measure metabolic activity by the so-called basal metabolic rate, which is the most common, the field metabolic rate for wild animals, or maximum metabolic rate on exertion, for example, for humans who are athletes training, under great physical pressure. So these are all very interesting concepts, but I will just focus on basal metabolic rate, which is kind of a standard. And it's interesting, these are the exponents beta physiological time, so the cycle time scales with the quarter exponent of the mass or weight of the organism. And the basal metabolic rates, most of it you'll find scale with 3/4 exponent. Sometimes for simplicity, 2/3 is used, which is close, 0.66 as opposed to 0.75, but 0.75 or 3/4 is the most common, with the exception of plants, which are quite often, they have an exponent of 2/3 or sometimes up to 1. And so that's in a simple form, scaling dependence, which you may see a similarity between the specific heat of solids, if that was the left-hand side, and the temperature, that would be the right-hand side, and an exponent for solids at low temperature would be exponent beta equals 3. So Lloyd Demetrius developed a theory which tries to find a general solution to this problem. What is the mechanistic basis of these scaling rules? Can we find something underlying the empirical observations, some sort of common principle? And then there are some additional rules, why variation between plants and mammals, and we'll come to it in a second, or maybe a minute. So this is of course about metabolism, and there are different ways of producing energy by different organisms. In bacteria, energy transaction takes place through bacterial membranes. In plants, through chloroplasts. And here we have direct link to quantum biology, because as I'll talk about it a bit later today, plants and some bacteria use these molecules to capture light, electromagnetic energy quanta, and use them for electronic degrees of freedom, which through a cascade of reactions lead to the formation of nutrient molecules such as glucose from CO2 and substrates. Sorry, glucose. In mammals, not exclusively, but most commonly, energy is produced by electron transfer chain reactions at the end of this cascade of reactions taking place in mitochondria. And in mitochondria in particular, there are 5 protein complexes in the inner mitochondrial membrane, which involve electronic transitions, hopping of electrons in an actually strictly quantum sense, that is coupled to proton movement across the membrane. So what do we have? Well, we have a range of geometries, units, organelles like mitochondria, in which chloroplasts, in which energy is being absorbed, transduced, and molecules produced that are used in other processes in living systems. And that's basically what we can say in a nutshell, but quantum metabolism theory of Demetrius integrates 3 aspects which are present. The first one is chemiosmotic coupling between electron transfer processes and ADP phosphorylation, which is the Mitchell theory. Nobel Prize winning chemist Mitchell proposed the explanation for oxidative phosphorylation. The second is actually critical, the storage of this metabolic energy in vibrational modes in molecular components of the membranes. And finally, the third aspect is that this vibrational energy, remember,